Good evening, all. Is it audible to you?
Yes, sir.
Hello? Am I audible?
Yes, sir.
Right. Yesterday we completed about microsporogenesis and male gametophyte development, megasporogenesis and female gametophyte development. Today the third unit, pollination. What is pollination? What is pollination? Just give me just a general definition.
Transfer of pollen grains.
Right, transfer of pollen grain from one place to the another place. That is called pollination. Clear. So here the pollination types. If you see, do you have book?
Yes, sir.
Hello? Right. So turn on page number 16. Types of pollination. See. Types of pollination.
Yes, sir.
Right. First, types of pollination. Xenogamy and gatinogamy. So of course, these two are the pollination types. What is xenogamy and what is gatinogamy? See in book, tell me.
Pollination takes place between two flowers of different plants, which is belong to same species.
Okay.
Next, geitonogamy. Pollination takes place between two flowers of same plant.
Okay.
Some consider this is a type of self-pollination.
Right.
Sometimes pollination does not take place in bisexual flower because of position of
Enough,
stamen and
enough, 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 enough. So
okay,
we
sir.
are discussing only two. One is xenogamy and gatinogamy. So let's discuss only these two only. First of all, let me share a slide. Just share this slide. Are you able to see this diagram? Hello?
No. Yes, sir.
Right. First of all, diagram C. Diagram C, xenogamy. Right. So xenogamy. So in xenogamy, pollen transfer between the flowers, different plants. This is important. Two different plants. So this is A plant different and this is B plant different. That is called xenogamy. And if you take the gatinogamy, in gatinogamy, same plant is same. This is only one plant. One plant, two different branches. That means plant same, but the pollination takes place between the different flowers, right? You can observe here in diagram clearly what is gatinogamy and what is xenogamy. Is it clear first? Hello?
Yes, sir. Yes, sir.
Right. Right. And later, the next given about the self-pollination prevention. Just read about it. I will explain. The simple, there are the
Sir,
self-pollination
which one,
prevented
sir?
by hmm.
Which one?
After gatinogamy, the self-pollination
Paragraph.
prevention methods are given. Even bisexual flowers. Actually, what happened in bisexual flower? In bisexual flower, this is for example plant flower. This is anther and gynoecium. This is gynoecium. So what is self-pollination? In self-pollination means the anther reach the same flower. So that is the self-pollination. So this method is prevention due to some reasons. What are those reasons they given? See in a book. I will explain clearly. Also given that is the hercogamy, heterostyle.
Dichogamy and hercogamy.
Right, right.
Can I read, sir?
Yes, yes, that's it. Dichogamy and ergogamy, right? So what is dichogamy given here?
Stamens and gynoecium mature at different times.
Right, right. For example, this is the flower. This is the flower. In this flower, anthers are already matured. But when the time of the this anther matured, at the same time, the gynoecium not matured. That means that is not ready to receive the pollen grains. If it is not receive the pollen grains, is there any chance to fertilization?
No.
Sorry for inconvenience. Right. So dichogamy immature in different times. Is the immature maybe occurs in pollen, that means the stamens, or immature occurs in, hello, am I audible?
Yes, sir.
Right. Maybe immature occurs in gynoecium also. Right. So ergogamy. In ergogamy also self pollination prevented. These two, dichogamy and ergogamy, mainly occurs in the bisexual flower. Actually, what happened in bisexual flower? The bisexual should be in self fertilization, but due to some reasons, whether dichogamy or ergogamy, prevented. In ergogamy means what is given there?
Stamens and stigma of bisexual flower are arranged at different positions.
Different position. The best example already you know about that. Do you know the hibiscus flower?
Yes, sir.
If you see the hibiscus flower, if you see, just wait, where is pen? If you see hibiscus flower, for example, this is hibiscus flower, pollen tube, that means stem nodes are present here, and this is actually this is gynoecium, and tip of the region, this is stigma. But where the surrounding sides, that means the lateral side, stamens are there like this. Where are the stamens now? Where are the stigma? Where are the stamens? Is it possible to self pollination?
No.
When the stamens matured, when the anther matured, when the anther dehiscence, these pollen grains released out, but this pollen grains falls down, but not reach upright region. Where are the stigma? The stigma above side, top region stigma are there, but the anther below the stigma. When the anther matured, when the anther wall break, the pollen grain released out and falls down, but not goes up. Due to this different position, self pollination prevented. Right? Is it clear?
Yes, sir.
Right. So here the pollination types important in short answer, and it is very easy. Very easy. Xenogamy, just when examination point of view, if they ask about types of pollination, first just given the definition, pollination definition, and one of the scientists Camerian studied about the pollen, that means pollination types, and here the two types of pollination, mainly xenogamy, geitonogamy. And later, in further, the self-pollinated flower shows the following two adaptation, dichogamy and ergogamy. Right? These are the most important. About next about the pollination takes place by abiotic and biotic agents. So you know the definition, the transfer of pollen from one place to another place, right? Hello?
Yes, sir.
Then
Hello.
how the pollen grains reach one place to another place? What are the agents? What are the vectors to transfer the pollen grain?
Wind, wind,
Right, wind,
water.
only wind. Oh, that's why here the classified the clearly two types, abiotic agents and biotic agent. What is abiotic? What is abiotic? In the name itself
Nat-
given abiotic.
Natural agents.
Biotic means
Nat- Natural agents.
non-living things. Simple. Abiotic means non-living. Okay. Just earlier you said about the wind, abiotic factor. And tell me other factors in abiotic agent to transfer the pollen grain one place to another place. Okay, wind. If the pollen grain transfer with help of the wind, that process called as anemophily. Remember it, anemophily.
Yes, sir.
And then tell me another agent, abiotic vector, wind. Okay,
Water.
right, water. If the pollen grain transfer with help of the water, that is called hydrophily. Hydro
Yes,
means
sir.
water.
Yes,
Okay,
sir.
wind complete and water and again and another one next. Of course, it is somewhat difficult. That is wind and water common.
Yes,
But apart
sir.
from that, rain also one of the best agent to transfer the pollen grains through the air. That is called ambrophily. So this is about abiotic agents. Abiotic agent first wind anemophily, water hydrophily, raindrop ambrophily. Simple. And next, what about biotic pollination?
Living organisms.
Just right, living organisms. What are those living organisms mainly involved in? Because
Insects.
generally
Birds.
we see the insect, birds,
Yes.
ornithophily, the
Insects
mostly
and
insect
mostly.
birds
Yes,
and
sir.
bats also. If the agent is insect, that is called entomophily. Agent is bird, that is called ornithophy.
Ornithophilia. Yes, sir.
Right?
Yes, sir.
Right. So like this, the pollen grains transfer one place to another place. Is it clear?
Yes, sir.
Right. Now, another important topic. This is also short answer. In third unit, all are most probably short answer. In first and second unit, already told you, first unit, microsporogenesis, that is for short answer.
Sir.
And male
Yes, sir.
gametophyte development, long answer.
Mm,
Second unit,
yes.
megasporogenesis, short answer. And female gametophyte development, monosporic long, bisporic long, tetrasporic also long. Right?
Okay.
Third unit, pollination, all are mostly short answers. Right? Let's see the pollen pistil interaction. What is pollen pistil interaction? Just give me the common general definition. What you understand about pollen pistil interaction? That mean the what I'm asking here, if examination point of view, the someone if the examiner ask about the pollen pistil interaction, you need to write some few points about that. Just give me just few words enough. Hello,
Yes, sir. I don't know, sir. I have no
what
idea.
is okay? What is interaction? Okay, so okay, where the pollen grains are deposited after during pollination, That means after a transfer one
Sir,
place to another place, where the pollen grains deposited.
on the other flower.
Right, exactly. But where in other flower?
Sta Stigma.
Right. So I am asking you, let me ask the simple question. The pollen grains, actually, for example, the hibiscus plants are there. And the hibiscus stigma receive only hibiscus flower, hibiscus pollen grain. Hibiscus stigma
Mm,
can receive only hibiscus.
yes. No, no.
Apart from the hibiscus pollen grains, other species, maybe the mangifera, neem, other type of pollen grains also deposited on stigma of the
Stigma.
hibiscus. But
Okay.
can the mangifera received by the mangifera hibiscus stigma?
No,
Can the neem?
no, sir.
Uh, just wait. Where is diagram? Sorry. Are you able to see the pollen pistil interaction slide? Hello?
No, sir.
Just wait. Where it is? So not clear actually. Where it is? Where is slide? Where it is? Just wait. Image flowers. Image file. Where it is? Wow. Now able to see pollen pistil interaction?
Yes, sir.
Right, but the words are not clear, I think. So anyway, I will explain clearly. Right, pollen pistil interaction. Right. So we can observe here in this diagram. So this is the stigma, right? You know, this is stigma part. This elongated style, this elongated part called style, and this is called ovary. The large place, the part-like structure that is called as ovary. Okay. Hello.
Yes, sir.
Right. How many pollen grains on stigma? For example, if this is A, this is B, C, D, E, F, different type of pollen grain there. But how many germinated? Only two germinated. What about the germinated one accepted and remaining are rejected because they are the other species, but not same species. That's why they are rejected. The first of all, in pollen pistil interaction, first recognition of compatible pollen. So the stigma pollen has the ability to recognize the right type pollen That is the compatibility pollen, that is same species. Remember it. The compatible pollen means the same species pollen, that is called as compatible pollen. And another species non-compatible pollen. So if the simple remember the what is pollen pistil interaction. The pollen pistil interaction means the stigma pistil has the ability to recognize the right type of pollen. Compatible pollen, that is the same species, that is called as pollen pistil interaction. So other than the species, they can reject other species. So definition clear? Hello?
Yes, sir. Yes, sir.
So the interaction between the chemical compounds of the pollen and those of the stigma. After reach the stigma, between the stigma and the pollen wall, there is small chemical interaction takes place. Let's see the germination of pollen grain stigma growth. First, pollen grain reach the receptive of stigma of pistil by act of the pollination process. Next step, pollen grains after getting attached to the stigma, absorb the water and swell. That is the second step. What is the first step? First step. In first step, not only the compatible pollen, the non-compatible pollen also residual stigma. Later, only the compatible pollen, the receive the moisture place and water from the stigma and slightly swell, observe water and slightly swell. The next step, pollen grain germinates. After swell, the next step is germinate and the stigma to produce pollen tube through one of the germ pore. Already we know the pollen grains have the number of pollen germ pores. Through the germ pore, what happened here? The pollen tube develop. This pollen tube from the pollen grain moves into the pollen tube, and that is tube that enter into the style. Right? This is style. This is style. After germination, the pollen tubes penetrate the style layers and penetrate towards the ovule. The later what happened? Just the last day that means yesterday we discussed about after pollen tube formation. Inside the pollen tube, two male gametes are developed. Two male gametes develop. So after the two male gametes, one of the male gamete fuse with female gamete or egg as a result formation of zygote and further developed into embryo. Right?
Yes,
So
sir.
actually, after formation, the male gamete, it enters the embryo sac through the filiform apparatus, one of the synergids, and fuse with the female gamete. As a result, formation of zygote and which developed as an embryo and finally settled as a seed. So this is about the pollen pistil interaction. Is it clear? And also, actually, in our book not given, apart from the some chemical reaction between the pollen grain wall and the stigma, some of the genes also involved. Some of the genes, the same genes mean the same species genes also involved to germination of the pollen tube or pollen grain. Draw this diagram. Pollen pistil interaction diagram. Draw it. Or take it. Take a snap, screenshot. Right. Is it clear? Hello.
Yes, sir.
Right. Next. Double fertilization. Just wait. Double fertilization very very important for short answer. Sorry, long answer. Fertilization after pollen pistil interaction. After pollen pistil interaction. Next, fertilization. That means After formation of male gamete, what happened later? Fertilization takes place. Let's see the fertilization. Already you know what is fertilization,
Fusion of male and female gametes.
right? And here the fertilization, the normal fertilization and double fertilization also there. Actually, I think before the fertilization, the how the pollen tube enter into the ovule. Just now we discussed about the pollen pistil interaction. After that, after formation pollen tube, this pollen tube how it is enter into the embryo sac. This enter may be the micropyle or maybe chalaza, may be the integument. In our book, clearly given the entry of the pollen tube.
Yes, sir.
The
Chalazogamy.
pollen tube, right? Porogamy in our book, the page number 20.
Yes, sir. Mm.
Page number 20. Can I show the diagrams or is it okay?
Okay, sir. No
Right,
need.
right. So the pollen tube of pollen grains enter into the ovule in
Mm.
various way. They are porogamy number one. Second one, chalazogamy, mesogamy. No need to show the diagram because the name itself being clearly. What is porogamy? What is poro means? The enter of the pollen tube into the embryo sac through the.
Integuments.
No, no.
Mm. Porogamy. Micropylar end.
Right. Through micropylar end.
Yes, sir.
The micropylar, that is pore. Micropylar is the pore.
Mm.
That is porogamy. What is second one?
Chalazogamy.
Chalazogamy means
Through chalaza. They
through
enter through.
chalaza. What is mesogamy?
They enter through integuments.
Through the integuments. So
Yes, sir.
enter into the integument, that is called.
Mesogamy.
That is called mesogamy.
Yes, sir.
Right? Is it clear? So
Yes, sir.
after anyway, the any direction, either porogamy, mesogamy, or
Chalaz.
chalazogamy, after anyway, any direction, the pollen tube enter into the embryo sac, right?
Yes, sir.
What happened after enter into the embryo sac later? Fertilization takes place. Here the fertilization type is double fertilization. Remember double fertilization. What is double fertilization? What is first? You know the fertilization process.
They fuse with two
This
eggs.
uh, somewhat right. Okay, 1 minute. I will show the. Where is diagram? Let's wait. Just wait. Where is the diagram here? Wait, my diagrams. Are you able to see the diagram? Hello?
Yes, sir.
Right. Show me, based on this diagram, just you can explain what is double fertilization. In diagram, two diagram clearly given. Hello?
Yes, sir.
In first release of sperm, that means pollen grain, male gamete
The
fused
first
with one,
male gamete fuse with
uh,
one egg cell,
right.
and next one the main nucleus.
No, secondary nucleus, here. Actually, if you see in this tube, two male gamete, blue color, are you able to see the blue color dots?
Yes, sir.
Right. So one of the male gamete fused with female
Egg cell.
gamete, egg cell,
Mm,
and rest of the one fused with ear. So another blue color dots at secondary nucleus, right?
mm.
Do you know the
Sir,
embryo sac? Embryo sac
yes,
structure.
sir.
Just wait, I will draw
Yes,
clearly.
sir. Antipodals and synergids and
Right.
nucleus.
So secondary nucleus, the rest of the one remaining male gamete fused with secondary nucleus, right?
Okay, sir. Yes, sir.
So here the fertilization takes place two times. One is the one male gamete fused with female gamete, that is the first one. And
Yes,
second
sir.
one, the remaining another one fused with secondary nucleus. Okay?
Okay, sir. Okay, sir.
Right. Just wait. So what happen if the first male gamete fused with female gamete? What are formed?
Zygote.
Zygote. And if the another male gamete fused with secondary nucleus, what is formed?
Mm, embryo.
After formation zygote developed into embryo.
Yes, sir.
Secondary nucleus, already in diagram given the name also, second diagram. After secondary
Endos
nucleus, round up structure. What is name given
endosperm.
next? Endosperm, right. Endosperm formed. 1 minute. Meantime, draw this diagram. Okay. Sorry for inconvenience. Right. So double fertilization clear. Are you clear?
Yes, sir.
Right. So third unit also completed. What is next? What is given fourth unit?
Endosperm.
Right, endosperm. So actually, where the one what just one is the important one. You know the concept, that is the first in double fertilization, first one male gamete fused with female gamete, right?
Yes, sir.
So that is called as syngamy. Remember it.
Okay, sir.
That is called as syngamy.
Okay.
And the next one, the next female gamete, sorry, next male gamete fused with secondary nucleus. That is called
Triple fusion.
triple fusion. So the syngamy and triple fusion mainly due to the syngamy zygote formed which is developed as a embryo. Due to triple fusion air formation primary endosperm which is developed as a endosperm, finally endosperm, right?
Yes, sir.
Syngamy discovered by Strasburger, triple fusion discovered by Navashin. So
Okay.
anyway, the first fusion takes first syngamy occurs. It follows the triple fusion. But after these two events, the first actually the formed endosperm instead of the zygote, instead of the embryo, instead of embryo development, even second time the fertilization takes place, but the endosperm formed earlier than the embryo. Right? Remember it. Of course, first syngamy, next triple fusion. But the earlier development endosperm and next follow the embryo. Why the endosperm earlier developed?
To provide nourishment to the
Right,
embryo.
to provide the nourishment to the developing embryo.
Yes,
That's the
sir.
first endosperm formed. Let's see the endosperm. Let's see the endosperm types. Just open the page number. Okay, are you clear about the endosperm? I think endosperm clear formation. And you tell
Yes,
me the pleidy of endosperm.
sir.
Pleidy endosperm which is applied or diploid or triploid.
Diploid.
No. Secondary nucleus pleidy in embryo sac or female gametophyte. What is the pleidy of secondary nucleus? Diploid.
Diploid, yes, sir.
Secondary nucleus diploid. After fusion with male gamete, it is turns into
Haploid.
no. Diploid 2n. Male
Hmm.
gamete pleidy. First male gamete pleidy. Only male gamete pleidy.
Diploid. 2.
Okay. How the male gamete developed? Male gamete development from? How the male gamete developed?
Stamens.
From stamens where?
Anthers.
From in anther where?
Micros. Mm,
Right.
microspore.
Exactly. Okay. From
Microspore.
microspore. You tell me the pleidy of microspore. Okay. How the microspores are developed inside the anther? By which division I am asking?
Meiosis.
Right. Exactly. After meiosis,
Mitosis.
the microspores are formed, which is
Uh,
meiosis. After meiosis, the pleidy number decrease or increase.
it undergoes mitotic divisions, sir.
As you said earlier, that is correct. Don't get confused here. What I'm saying here, first of all, the microspores are developed from inside the anther, right?
Yes.
You said the meiosis division, right? Okay, remember simple thing after meiosis, wherever the meiosis takes place, automatically the after as a result of meiosis, the upcoming cells are always haploid cells.
Okay, sir.
So now tell me the microspores are haploid or diploid?
Haploid.
Haploid, right? So from the microspore, male gametophyte developed and male gametes are developed. Now tell me this male gametophyte, male gamete by mitotic only, not meiotic.
Okay,
Mitotic means the chromosome number neither decrease nor increase. So you tell me the automatic final. No, that means the male gamete pleidy.
haploid,
Microspore
sir.
haploid. By
Yes, sir.
mitotic only, male gamete formed.
Yes, sir.
Now tell me the pleidy.
Haploid.
Right, haploid. So now the male gamete haploid. Okay. Male gamete. Oh, just wait. Right, male gamete haploid. But in secondary cells, just wait. Let me draw. Hello?
Yes, sir.
Right, right. Just wait. What happened here? Whiteboard, whiteboard. All right, share. Simple thing. Embryo sac, secondary nucleus. It is 2n, 2 nucleus. So it is 2n already. Female gamete, n. Synergids, sorry. Right, okay, synergids. And antipodals, 3. So already it is 2n, right? In pollen tube, 2 male gametes are present. Just we have seen the in diagram. One of the male gamete fused with egg cell because here the egg are applied or male gamete also applied. As well diploid embryo, zygote formed which is developed embryo. And this applied male gamete, rest of the applied male gamete fused with secondary nucleus. Secondary nucleus, right? Now tell me here already Suhana, don't draw. Right, thank you. So after formation, the secondary nucleus already it is diploid. Now the applied male gamete fused. Now it is developed into, it is diploid secondary nucleus and haploid nucleus. Now finally it is 2n plus 1n. Fusion 1 plus 2, 3n. 3n primary endosperm formed. Now tell me the endosperm ploidy. Hello? 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 Hello?
Yes, sir.
Right. Now tell me endosperm ploidy.
Haploid.
Uh,
Sir, I'm not getting, sir. Once explain me.
just wait. I will try. Right. Are you able to see this diagram? Hello? Are you able to see the whiteboard diagram? Hello? Hello? Hello?
Yes, sir.
Right. So this is embryo sac. Do you know the embryo sac structure? In embryo sac, how many cells are there? 7 cells, 8 nucleus. The 7 cells are egg, first one. Either side synergids. So, and the three antipodal cells and center secondary nucleus. Here the each and egg cell haploid and this synergy haploid, antipodals also haploid. As it in middle, two nuclei present, these two together, n plus n, it is already 2n. Diploid state. Secondary nucleus diploid state, right? Hello?
Yes, sir.
Right. So, what happened after formation pollen tube? Let's change the color. Pollen tube. Inside the pollen tube, how many male gametes are there? Two male gametes are formed. One of the male gamete fused with egg cell female gamete, already female gamete N. The male gamete also N. As a result, formation as a diploid 2N and which is developed as a zygote and developed into embryo. And another remaining N male gamete, haploid male gamete fused with secondary nucleus. The already secondary nucleus 2N and fused with haploid male gamete. Now as a result formation of 2N plus N.
Triploid.
Triploid. Now which is primary endosperm and further developed into endosperm. Simple. Right?
Okay, sir. Yes, sir.
Right. So now let's see the types of endosperm. The simple, very easy, damn easy. So here the cellular endosperm. One is cellular endosperm and another one is nuclear endosperm, helobial endosperm. Three types. The diagram itself we can understand. What is first one? Cellular. The name given is cell. Second, free nuclear endosperm. And elobial, that means the both together. Simple, let me draw here. Are you able to see this diagram? Hello?
Yes, sir. Yes,
Right.
sir.
Right, just wait. Another, let me take another board. First, let cellular type. Cellular type. Already you know the process of endosperm formation. Just I directly start up formation of primary endosperm. After primary endosperm formation, the mitotic division takes place. As a result, inside two cells are formed. And again further divided, 4 cell further divided, 8 cell further divided. Yeah, 16, 32 like this, uncountable number of cells are formed. So that means first nuclei form. After formation, the nuclei, number of nuclei formation, what happened here? The cell wall formation around the nuclei. Around the nucleus cell wall formation. So now it is developed into cell type. So this is called cellular type. This is called cellular type. Simple. And second type, nuclear type. The name itself, nuclear. That means free nuclear formation. So after formation here, development, only the free, there is no septa. There is no septa formation between the one nucleus to another nucleus. The free nuclear, freely, these nucleus are freely formed. So this is called free nuclear. This is called free nuclear development. Right? Is it clear about, are you clear about the cellular and the free nuclear?
Yes,
Hello?
sir. Yes, sir.
Cellular simple, the formation same. But after formation, number of nuclei at the cell wall formation around the nucleus in cellular
Nucleus.
type. But
Yes,
in
sir.
free nuclear, no cell wall formation. Only the nuclears are developed. In this, the free nuclear, the best example, the endosperm appear in liquid. Cellular type, the endosperm appeared in solid. The best example, coconut. Do you know coconut? Hello?
Yes, sir. Yes, sir.
In coconut, what you drink in the tender coconut, what you drink? Water, right?
Yes, sir.
So the water is the free nuclear endosperm. When it is matured, when it matured, When it matured means the matured coconut we offering to God. So in this, the what you find out in general after break it out, the cellular endosperm, that is what we eat, cobra. Hello,
Meso.
right. That is the cellular type,
Okay, sir.
cobra. That is cellular type. The water only liquid. That is nuclear type. So
Yes,
now
sir.
the last one, the holobial. Holobial, simple, some in endosperm types, half of the cellular and half of the free nuclear. The both mixed cellular and free nuclear both mixed together. That is called as a holobial type. And Hastoria formation. Anyway, any type, whether it is cellular type or free nuclear type, Hastoria form. Hastoria, the elongated projectors are formed from the endosperm and which is penetrate to into the developing embryo. Right? Hello?
Yes, sir.
And also which is received the nutrition from the surrounding cells, surrounding new cellular cells. They receive the nutrition and also send nutrition to the developing embryo. That is the function of Hastoria. So this is all about the endosperm types. Endosperm types. This is very, very important for long question and very damn easy and important. So the endosperm Hastoria also important. The Hastoria endosperm occasionally produce the special structure as the embryo sac whole. These have the morpho-histogenic organizations which is from the remaining endosperm. The Hastoria in nuclear endosperm, free nuclear, and the Hastoria in holobial endosperm, Hastoria in cellular. The same. Already, we are just now we discussed about the cellular and holobial and free nuclear. So now the endosperm function, what is the function of endosperm? What might be, you just assume,
Which give complete nourishment and nutrients to
right?
the embryo.
Right, endosperm contain the growth regulators and nutrients and which is useful to developing embryo from the zygote and also involved in seed germination. That means seed germination needs some energies, needs some nutrition, which is provided by the endosperm, right?
Yes, sir.
The various substances like carbohydrates, proteins and fats and enzymes, vitamins, growth attractors are formed inside the endosperm. Growth regulators such as indolestic acid, IAA, gibberellic acid, GA, nicotinic acid, xanthine, they are formed. All these substances interacted with the growing embryo and cause development. So this is all about the endosperm. Just in endosperm, simply just the formation, types of endosperm, and write about Astoria. This is about endosperm. Is it clear?
Yes, sir.
The next another important topic, embryo. How the embryo formed?
Zygote turns into
Right,
embryo.
zygote. The single cell zygote developed into embryo. So embryo also dicot embryo development and monocot embryo development. The dicot embryo development somewhat different with monocot embryo development. Before that, you see compare the difference between dicot embryo and monocot embryo. In the dicot embryo development and structure, long essay, important one, and monocot embryo development and structure also long essay, very, very important. We will complete tomorrow about this both. Okay?
Okay, sir.
Right. Any doubt today class?
No, sir. Sir, this question, sir.
Which one?
Endosperm Hastoria.
Yes, endosperm, Astoria also, the types of endosperm. This is the come under
Sir,
the types of endosperm.
we have to write whole, sir.
Whole, right,
Types of endosperm and
right, types of
endosperm.
endosperm and also Astoria.
Okay, sir.
And also you mentioned the functions also.
Okay, sir.
Right.
Okay, sir.
Thank you,
Thank
one and all.
you. Okay, sir.